What are those, sir?
Those are diphtheria.
Tell me more about it, please.
I have book about it. You'd better read it.
Diphtheria is a serious communicable bacterial disease that causes severe inflammation of the nose, throat, and windpipe, or trachea. It is caused by the bacterium. The name of bacterium is Corynebacterium diphtheriae. The bacteria produce toxins that cause an abnormal membrane to grow in the throat, which can lead to suffocation. Other dangerous complications include paralysis and heart failure if the toxins spread throughout the body. Around 10% of people exposed to diphtheria die from the disease. Corynebacterium diphtheriae is a pathogenic bacterium that causes diphtheria. It is also known as the collapsed Loeffler bacillus because it was discovered in 1884 by German bacteriologists Edwin Klebs and Friedrich Loeffler. Corynebacteria are gram-positive, aerobic, non-motile, rod-shaped bacteria classified as actinobacteria. Corynebacteria are related phylogenetically to mycobacteria and actinomycetes. They do not form spores or branch as do the actinomycetes, but they have the characteristic of forming a regular, club-shaped or V-shaped arrangement in normal growth. They undergo snapping movements just after cell division, which brings them into the characteristic form resembling Chinese letter. The best known and most widely studied species instead of other genus Corynebacterium is Corynebacterium diphtheriae, the causal agent of the disease diphtheria. The best known and well-studied bacterial protein toxin is diphtheria toxin, or DT, produced by Corynebacterium diphtheriae. DT is a bacterial exotoxin of the A-B prototype produced as a single polypeptide chain with a molecular weight of 60,000 daltons. The function of the protein is distinguishable into two parts. Subunit A, with a molecular weight of 21,000 daltons, contains the enzymatic activity for inhibition of elongation factor 2, or EF2, involved in host protein synthesis. Subunit B, with a molecular weight of 39,000 daltons, is responsible for binding to the membrane of susceptible host cell. In vitro, the native toxin is produced in an inactive form which can be activated by the proteolytic enzyme trypsin in the presence of thiol, a reducing agent. The enzymatic activity of fragment A is masked in the intact toxin. Fragment B is required to enable fragment A to reach the cytoplasm of susceptible cells. The C-terminal end of fragment B is hydrophilic and contains the terminants that interact with specific membrane receptor on sensitive cell membranes, and the N-terminal end of fragment B is strongly hydrophobic. The specific membrane receptor for the B fragment has recently been shown to be a transmembranous heparin binding protein on the susceptible cell surface. DT enters its target cells by either direct entry or receptor-mediated endocytosis. The first step is the irreversible binding of the C-terminal hydrophilic portion of fragment B to the receptor. During the receptor-mediated endocytosis, the whole toxin is taken up into an endocytic vesicle. In the latter, the pH drops to about 5, which allows unfolding of the A and B chains. This exposes hydrophobic regions of both A and B chains so that it helps the T domain insert into the vesicle membrane. The result is exposure of the A chain to the cytoplasmic side of the membrane. There, reduction of proteolytic cleavage releases the A chain in the cytoplasm. A is released as an extended chain but regains its active globular conformation in the cytoplasm. The A chain catalyzes the ADP ribosylation of EF2, the toxin or subunit A intoxicates the cell by utilizing NAD as a substrate to catalyze the attachment of the ADP ribose portion of NAD to EF2, which inactivates EF2 function in protein synthesis. Bacteria can live in the mouth, nose, throat, or skin of infected individuals. Diphtheria is most commonly spread when someone ingests or inhales the cough or sneeze droplets from an infected person. Symptoms occur between 2 and 10 days following infection. Occasionally, transmission occurs from skin source of through article soil with discharge from the source of infected person. Sometimes a person has such a mild case of diphtheria that they don't realize they are sick. However, they are still contagious for about 6 weeks and may infect a lot of other people. An apparently healthy person who spreads an infectious disease is called a carrier. Signs and symptoms of diphtheria may include runny nose, severe sore throat, fever, generally feeling unwell or malaise, swollen lymph nodes in the throat, a furry gray or black coating on the throat membranes, which is made up of bacteria and dead cells, breathing problems, swallowing problems. Sometimes diphtheria causes a skin infection. The wound is sore, inflamed and full of pus, and may be surrounded by grayish skin patches. This condition is known as cutaneous diphtheria. It is quite rare in developed countries. Toxin production of Corynebacterium diphtheriae occurs only when the bacillus is self-infected by a specific virus or bacterial page carrying the genetic information for the toxin or tox gene. Only toxigenic strains can cause severe disease. Diphtheria toxin production is encoded by a bacterial page carrying the tox gene that gains entry into Corynebacterium diphtheriae. The toxin consists of an A fragment and a B fragment, and the B fragment binds to the receptor or heparin binding epidermal growth for precursor on the cell surface, and the whole molecule is then taken into the cell by endocytosis. In the closed environment of the endosome, acidification occurs. The low pH level causes the B region to unfold and form a membrane channel, allowing the A domain to pass through the membrane into the cytoplasm. The disulfide bond linking the A and B regions is produced, and the A subunit is then freed to bind to ADP ribosylate elongation factor 2 or EF2. Ribosylation interferes with the ability of EF2 to add amino acids to a peptide chain, blocking the protein synthesis and causing cell death.